the keynote speaker for this meeting. There is the short introduction. He's a leukemia doctor that doesn't do leukemia anymore. Or I can introduce Craig Thompson as the Benno Schmidt Chair of Cancer Research at Memorial Sloan Kettering, and a physician scientist who really helped shape modern cancer biology, immunology, and metabolism. Between 2010 and 2022, for these 12 years, he was the president and CEO of Memorial Sloan Kettering, where it was a period of major growth in immunotherapy, precision oncology, genomic, and cancer care. Scientifically, Dr. Thompson is known for many fundamental contributions to understanding programmed cell death, apoptosis, immune cell activation, and metabolic pathway that regulate cell growth and survival. I can go another 10 minutes, but we're running out of time to tell you exactly his contribution in immunology, apoptosis, metabolism. I'm not going to let him in, but also most important, he told me his mentees, the people that went, his legacy, and contributed and still and continue to lead major cancer program. To us, Matthew van der Heijden, who is a Dana-Farber physician, GU oncologist, but the director of the Cancer Center at MIT, and my dear Ziad Bakouni, who was my postdoc and now in your lab, a fellow at Memorial Sloan Kettering. And since Dr. Thompson, is already on stage, here you go, Craig.
Thanks. Well, I thank Tony for that very kind introduction. It's unusual for a meeting like this of the world's specialists in kidney cancer from all the disciplines that are responsible for taking care of patients and the patient advocate community to be inviting a general oncologist into this audience. Since for the last 15 years as president of Memorial Sloan Kettering, I was the privileged, the only employed general oncologist because there was always somebody better than me to take care of a patient in need. And so it's really a pleasure to be here and span this transition that we have from the omics era that Tony just talked about to this exciting new possibilities of what AI will bring to our use of clinical databases to optimize therapies, which is what the rest of the morning is going to be about. So I'm not going to talk at all about any of that kind of stuff. I have no expertise in AI. All of the expertise I have in AI, Tony just told you about, it's his common mentee. We're all now in our laboratory-based efforts have computational biologists, and he trained an amazing scientist, Ziad El-Bakouni, I'll mention him again in a minute, and has brought us into the modern era. But I want to remind you, so the only real important thing to remember for the next half hour is that AI is only as good as the data sets you train it on. So we have amazing data sets about genomics. We have amazing evolving data sets about proteomics. What I'm going to try and convince you is that there's more data sets. More of it has to be single cell, you already know that. But in addition, metabolism plays a huge role. And that pioneering insight came from kidney cancer, and so I'm going to tell you that story starting in 1863. And that's what I'm going to do for the next half hour, okay? So for background, I have to give you the disclosures. Those are all the disclosures. You can read them at your leisure. They have nothing to do with what I'm about to tell you about. All of the work that I'm going to tell you about was done by the following individuals in my laboratory. They're the three at the top. Ji-Young Kim, who is the person who did all the experiments I'm going to show you, but he followed up on a medical oncology fellow who was a nephrologist, who was training, who wanted to ask very specific questions about the metabolism of kidney cancers. And she taught us, and that's Aggie Bielska, who she's now here in Boston working for in a translational research program. But she passed on the project to Ji-Young Kim, and it's been joined, as you just heard, by Ziad El-Bakouni, who will give a talk at the end of the day about the really exciting new stuff we have learned from the omics-based approach, particularly around the importance that has been very known about why so many kidney cancers initiate with VHL or mutations, as has been discovered in now multiple data sets of mitochondrial DNA. So you'll hear about that if you want to hear about that part of the story, which I'll briefly touch on from Ziad this afternoon. So why did my lab get back and interested in kidney cancer? It's not like you needed any extra help. We have a tremendous community devoted to kidney research. It happened during the pandemic, and it happened in the pandemic because Ari Hakimi came to my lab, which he regularly did to advocate for our kidney cancer group at Memorial Sloan Kettering. He came to my office and he said, you know, there's this amazing thing, and you should understand the metabolism of kidney cancer because there's this obesity paradox. Now, at the time, all of us have learned over the last 20 years that cancer is a huge risk, obesity is a huge risk factor for cancer. And that's true of kidney cancer as well. And so your likelihood of getting cancer has doubled as our obesity has doubled in this country. But the remarkable fact, and it was learned by this community, so again, this is the slide that got me interested from Ari's sort of meta-analysis, the team of people that did that, and Ari was the senior author, and that was published in the Lancet Oncology. And this is to get at what Brian said. Patients really care about better outcomes, about making it to that next milestone. And patients at the initial diagnosis, if they are obese, they're morbidly obese, they have a better outcome. They're more likely to make that next milestone. They're more likely to be long-term disease-free independently. And that, as you know from your community, has cut across precision therapy databases. That has cut across immunotherapy databases. There's something fundamental about kidney cancer that makes obesity a good prognostic factor. And that, to us in the metabolic community, is pretty interesting because everything else about obesity is that it increases inflammation, increases all these issues across the panoply of cancers, but uniquely in kidney cancer, it has a good outcome. And that's better than any precision therapy or immunotherapy in terms of outcome that you've actually seen or will talk about at this meeting in terms of difference in long-term survival. So I'm going to take you back because most of the last 20 years is at Memorial and in many other places, we've all been looking at genomic databases and we've been looking for biomarkers. Those have been the two really big innovations in cancer across the board. But the earliest biomarker in cancer is actually the renal clear cell. 1863, pathologists realized that cancer was a separable disease from infections in the kidney because there was a unique morphology to the cells that were transformed. And I don't need to tell anybody about that. It's just here on the slide. You can see that unique clear cell morphology. These large syncytia of cells that are a clear cytoplasm. They don't appear to have cytoplasm, and they're surrounded by this vascular network. And so these are the highly vascular renal clear cell carcinomas you have all worked on. The most common form of kidney cancer. It is pathognomonic of the disease, and now you think of it as synonymous with VHL, and I'm going to try and tell you that's not actually true. And so that 10% that you don't see VHL mutations are telling us something much more fundamental about cancer than we ever believed before, at least that's the way we're beginning to think. So this is dropped off as a biomarker. The community doesn't really care very much about it. The community that rescued the understanding of what this was the community of cardiovascular researchers, which are asking, how does the heart always get enough energy to beat so that it never will fail to beat the next time you need it for 90 years? Okay, and so what they did is they said, what is in these cells? And it's not just kidney cells, it's every cell in our body. What those cells are filled up with is lipids. Those are all lipid droplets that are crowding out everything else in the cell. The cell has basically become a reservoir in renal clear cell carcinoma of, in fact, these lipid droplets. The lipid droplets themselves are confined in most cell types purely of triglycerides with just a little bit of cholesterol. And I'm not going to talk about the architecture of those things because what's different about the renal clear cell carcinomas is they fill up not just with triglycerides. In fact, that's a minor component. They fill up with cholesterol esters. And cholesterol esters are interesting. Nobody in this room pays attention to them. They're really part of the liver disease world because that's how you take cholesterol esters and deesterify them to initiate cholesterol into biosynthesis for removal of cholesterol. So why is the kidney cell accumulating these, and what good is it to the cell? It's pathognomonic, it's a biomarker, and it makes no sense. And why Ari got us interested in this was our lab has spent a very long time, as Tony just said, interested in how the body partitions nutrients to the different cell types so they can do their job. And along with that, cancer is the disease of that. How does the cancer become cell autonomous to steal the body's resources to fuel its own growth and proliferation? And what we discovered 25 years ago across all the lineages of our body is it's growth factors. It's not rocket science to understand this. Growth factors distribute our body's resources. Receptor tyrosine kinase, including all the cool things it does to initiate new transcription and translation that you've heard about, also initiates the uptake of essential nutrients to engage in growth and proliferation, and that's what's shown in this slide. Growth factor signal transduction engages cells in this process first described by Otto Wilberg called aerobic glycolysis, in which cells take up more nutrients than they need of all major classes and builds its new daughter cells. And it does that by building lipids, by building nucleic acids, and building proteins to duplicate everything in the cell faithfully. And you don't, so this is a really important paradigm to understand that we've learned over 25 years. Cells don't build their daughters. It's the most precious thing they do during wound repair or tissue replacement from what you ate for lunch. They build it from the elemental precursors, the amino acids, the glucose, and they take that carbon and nitrogen and they build everything de novo so that they can do that. And that's regulated by growth factor signal transduction and the lineage-specific receptor tyrosine kinases, plus the various common growth factors that are hormones like growth hormone and other are what mediate that process. In cancer, when we studied it, what we discovered, along with a very broad community across the whole cancer space, is the most common initiating mutations that occur across the entire cancer spectrum are either lineage-specific receptor tyrosine kinases, you know all the stories, or activating mutations of PI3 kinase or AKT, which are the two critical kinases, one a lipid kinase, the second a serine threonine kinase, that take receptor tyrosine kinase signaling into the world of metabolism and permit the cell to take up cell autonomously glucose and amino acids. I'm not going to talk about that. And from that, cells build all their building blocks. So immediately when we started realizing this about 15 years ago, kidney cancer stood out. We collaborated with Celeste Simon and others, Bill Kalin, to try and understand why kidney cancers were so different. Because they don't have, the tumors you all take care of don't initiate with the receptor tyrosine kinase mutation, and renal clear cell of that subtype that's most common is the least likely to do that of the different subtypes that you're working on. And so I want to just take you through very quickly what we thought was going on and everything else, and that was that receptor tyrosine kinase through it, PI3 kinase and AKT controls glucose uptake, and this is just to do the fatty acid part. I'm not going to talk about the nucleic acids or the proteins. That would get us into hours later. What happens in this signal transduction pathway that's so commonly mutated in other cancers is you initiate cell autonomous glucose uptake. That glucose floods the cell with more glucose and byproducts of glycolysis that it never needs. It does it at multiple steps. And it turns the glucose into a precursor, and the major precursor it needs to turn into is the production of fatty acids. And it does that because the initiating step of the TCA cycle in mitochondria is citrate production. And when the mitochondria doesn't need to make any more ATP because the cell's ATP needs are satisfied, it secretes that citrate into the cytosol to provide a source, as that citrate is broken down in the cytosol, of acetyl-CoA from which you build de novo all your fatty acids. All endogenous fatty acid synthesis comes from that citrate-derived acetyl-CoA, as we and others have shown. And that allows cells to build phospholipid bilayers. Now, there's a little side effect, which is lit in black. I made the arrow a little bigger for this talk. You still need some essential fatty acids, just like you need essential amino acids. And that's a little bit part of the story. The essential fatty acids we need are the ones that all the nutritionists talk to you about. They're the polyunsaturated fatty acids that we can't make, that we get from our diet, either from plants or from various other sources. And those things are what we build the signaling rafts that people talk about that are associated with lipids. And they build the prostaglandins and the arachidonic acid derivatives that are signaling molecules within our body. And so you do need a certain amount of essential amino acids, and it's always been a mystery what allows cells to take those things up. But when we looked among all the other cancers, what we discovered is it's primarily the LDL receptor that's responsible for bringing those on. So receptor tyrosine kinase signaling in L cells that we've studied up until we get to the kidney cancer actually are initiated by activating LDL receptor uptake of lipids, and that's how you satisfy your essential fatty acid needs. So we've been thinking about this problem for a long time. One of the nice things about this mutation that turns on what Warburg discovered in 1920 about aerobic glycolysis is it prevents cells from actually being terminal effector cells. So cells stay in a proliferative, capable, stem-like state as long as they're engaged in aerobic glycolysis. And the reason they do that is terminal differentiation isn't just gene expression patterns. So what I've showed you in the middle, one of the things that we and others have been collaborating on in the metabolic community is it's intermediate metabolites that accumulate during this process of taking up more nutrients that alter chromatin structure through epigenetics. The marks are acetylation, that acetyl-CoA that comes actually is an informative mark that we have nutrients and we can do gene expression. It's also demethylation to activate the genes required for differentiation. That's also regulated by metabolates in the TCA cycle, by the ratio of alpha-ketoglutarate to succinate that we and others have shown. And that allows cells to differentiate, to turn on a gene expression program that can then stably maintain in an epigenetic way. So you can differentiate into a specialized cell. In the case of this community, the proximal renal tubular cell. Why is that important? Because that cell can only do its job if it engages in oxidative phosphorylation. The vast majority of true effector cells in the tissues and organs you care about only do their job because they need ATP to do it. Your muscles to contract, your heart to keep beating, purely burns fat because it's a thing it can store and keep forever. Never bothers with the glucose, even though it sees the highest level. We still don't know why that works. Cardiologists have been working that for 80 years. But that's what the heart does. The brain also only burns lactate if it can, because it doesn't have to waste time with glycolysis, because all it needs is a membrane potential to carry out action conductions and to keep circuits going that live membrane. In the kidney, you've got to resorb all the solutes you've just filtered from the glomerulus and take them back in. You know that in the glucose case, it's SGLT2. That's why it's such an amazing therapeutic and always been overcome by the GLP-1s. And so that takes energy. It's purely an energy problem. How do we get enough energy to man the pumps, to get rid of water, to balance the salt solutions, and to keep the critical nutrients that have gone into urine from the glomerulus back up? That's the normal job of the kidney. One of the different things about this community, and this is the challenge when I get invited as a general oncologist to these kind of communities, is you don't often speak to the general nephrologists and talk about the diseases they talk about and understand why kidney is in that spectrum and what kidney teaches you about that organ itself. And one of the things that's most interesting for this talk is that they bumped into loss of fatty acids into the urine as the worst prognostic factor in nephrotic disease in the last 10 years. So lipids are also secreted out into your urine all the time. And I actually think that's a huge insight as we go forward with this process. So what does the kidney have to do with this and kidney cancer? Well, in this little model that I told you about metabolism, the one thing that's in common with the common mutations, both the genetic ones, the common one in VHL, and now recently, as you'll hear from Ziad this afternoon, mitochondrial DNA mutations all share in common is the inability of that cell to ever engage in effective oxidative phosphorylation. VHL prevents glucose from getting there by converting the cell to aerobic glycolysis on its own because of the properties of HIF-1 alpha as a transcription factor. As it activates HIF-1 alpha, that's why the therapeutics we just heard as a milestone are so important, because it blocks that ability of HIF-1 alpha to function in changing the metabolism. SDH and FGH completely destroy the ability of the TCA cycle to be coupled to oxidative phosphorylation. And yet they were known as genetic syndromes, right, to cause as their major end-of-life event kidney cancer when they were reduced to homozygosity, so they're tumor suppressors. The TCA cycle is a tumor suppressor. And more recently, the discovery now in almost every genomic data set as it's been looked at, mitochondrial mutations, particularly of complex 1, also do exactly the same thing. And they're reciprocal in their rates with VHL across the kidney cancer space, as Ziad may talk to you about tomorrow. And so one of the things that's interesting in this idea that cancer is a block to terminal differentiation and taking up your effector role is that, in fact, these mutations that initiate kidney cancer prevent the kidney cancer from actually taking its job as a proximal tubular cell. So all they know how to do is still proliferate, and there are growth factors around that continue to do that. So how do we get to this lipid droplet state in VHL tumors? Well, the normal role of the proximal tubular, as I said, is to take up nutrients that are being lost, and salts and water that are being lost in urine from glomerulus filtration. And for the fatty acids are mostly filtered out by the free fatty acids, which are relatively low in our body if we don't have any proteinuria in our urine, but they are retaken up. They're also taken up by the LDL receptor in the normal, just as I told you, the normal proximal tubular takes up by the LDL receptor fatty acids for the blood, and the cell uses purely, like the heart. After the heart, the proximal tubular cell is the second most common, after the heart, cell that depends on fatty acid oxidation for its bioenergetics and support. And that's its real role. Okay? And so the normal cell burns fatty acids to be able to take up the glucose and amino acids that are lost, bring them back to the bloodstream so you don't lose them, and to balance the salts. And the reason you store in the heart, because that's where it's been best studied, triglycerides in these things that are called lipid droplets is that so on a rainy day when you need more nutrients, this is the model, I stole it just from a recent review, those lipid droplets that are stored forms of triglycerides and cholesterol esters can be recycled by degradation either by lipolysis, which is what's on the right there, or by autophagy, by eating the lipid droplet in the lysosome and using liposomal lipases to release the acyl chains for beta oxidation, which is a really good fuel for oxidative phosphorylation. That's the story out in the community. So how did we, when we started to think about this, tie this to renal clear cell carcinoma? Well, the problem with this is that, as you know, in tissue culture, these cells don't really look like clear cells. And sometimes when they metastasize, they don't look like clear cells. We can talk about that in a question and answer later. But what's interesting is if you take the NIH-60 panel, the panel of cells that have been isolated by the federal government to give us all access to cell lines that are classic descriptions of the common cancers, the ones that are in the community of clear cell carcinomas, the CACI 1 and 2, 498 and 7860, which were part of the original NIH-60 panel back in 1983 when it was formed and have been cultured for 40 years, they do two things. When you do gene expression analysis, they still know they're proximal tubular cells. So they've actually differentiated epigenetically that program, and they maintained it in everybody's lab here for that period of time. They know that's what they are, but they know how to proliferate. And what they do is they have lipid droplets, but they're not dramatically better than every other cell. They're just a little bit more on the spectrum compared to other cells. And that's why we got disappointed as a community, and that is a marker. So we started to play around with that because we know growth factors can control increased lipid uptake. So we thought this would be cool. And the classic experiment that's done in a basic lab is to take serum away. The reason why serum is in tissue culture is it provides an array of growth factors that all cells can live on, including the IGFs and other kinds of factors that come, PDGF that is released from the degranulation of platelets that work on every cell. And so we said, oh, maybe the reason they lose that is they're not getting the normal signal transduction because they don't have receptor tyrosine kinase signaling. And so you do the simple experiment that's been done for 50 years in labs. You do serum deprivation. You just take serum out of the media and ask what happens. And so here's the story, and the picture's worth 1,000 words. So that's what 498, a classic renal VHL mutant clear cell carcinoma looks like. It's got lots of those lipid droplets. They're stained in green because it's a dye taken up by a neutral lipid environment. But if we serum deprive them over, as you can see in the pictures, 24 hours, those lipid droplets disappear. By 48 hours, you can't see them. And you can quantitate that over there, which says that there's something in serum that's maintaining them, and when they don't have whatever's in serum, these droplets are recycled somehow. presumably into this lipolysis pathway. So we did what everybody did. The textbooks say it's supposed to be beta oxidation, but kidney cells, as best we can tell, don't do oxidative phosphorylation. VHL keeps that from happening, so we sort of tested that. What gets released from triglycerides when they undergo lipolysis are long-chain fatty acids. The only way they get in to the mitochondria to be burned is through this shuttling system called CPT1 and 2. We can eliminate genetically CPT1 and A and 2, and the droplets still go away, and they never come back if we serum deprive. And we've done many other experiments that say this is nothing. They never burn. Kidney cells never burn these fatty acids as part of this process. They're storing them, but they're not storing them because they need them as a source of energy. They already have aerobic glycolysis going on because of VHL. So what can you do? Well, you can say, is it really serum? So that you can do the opposite experiment. You can just add more serum. Now, I should tell you that if you add more than 50% toxin, because remember, serum is clotted blood, right? It's the filtrate of clotted blood, so there's lots of activated kinds. So if you go more than 50%, you just get unhappy cells. But what you can see is when we increase the amount of fat of bovine serum that's present in these media, the lipid droplets get bigger. That says there's something that's causing kidney cancer cells to accumulate these fat droplets that are pathognomonic for them if you put them in culture that comes from serum. And now I'm going to save you a whole lot of biochemistry and all kinds of experiments and just show you, here's what we discovered. We took all the guesses. We're guessers. So we guessed all the favorite growth factors. None of them made a difference. All the cells died. Except we decided to add one last thing. There are lipids, actually, in the extracellular environment. And it turns out if you just add oleic acid, a monounsaturated fatty acid we make as the preferred lipid we put into lipid bilayers, you fully not only keep the cells from dying in response to serum deprivation, they now grow just as well as if they didn't have the serum. The only thing they need from serum is that oleic acid, and then they're off to the races. And they double every day for as long as you want. So these cells can't make enough endogenous fatty acids to maintain their biology as a transformed cell. They have to have it in trans, but if it's in trans, they can take it up and use it. So is that meaningful? Yeah, so here's just a simple experiment. Serum-deprived cells, if you just culture along, they'll hang in there, but they'll ultimately die over a number of days. But if you add oleic acid, which is what the O is for the SSO, they proliferate beautifully. And in complete media, both cell types show. It doesn't matter whether you add oleate or not. As long as you give them complete media, they can take up what oleic acid is there and replace that, or whatever lipids are there. So it's what I told you about. What we supply in serum on the right-hand side, I'm going to skip the synthesis because I sort of went over it already. Normally, you make a phospholipid bilayer from adding to glycerol 2 acyl chains, that's what's on the right, and you put a polar head group, which is either preferably choline, but sometimes phosphatidylserine. People may know about that, and it's really important you get it right. You need one saturated fatty acid, which is always in the SN1 position, and you need one desaturated fatty acid, which provides flexibility to the membrane because that kink that's created by the double bond actually gives flexibility to the bilayer from which these phospholipids are built by. And you can put either a monounsaturated fatty acid, which is what we do naturally, into this membrane, or you can put polyunsaturated fatty acids we get from our diet. And serum contains all three. So we just said to do the simple thing. Let's take the 3 components that are known, which are saturated fatty acids, monounsaturated fatty acids are naturally produced, we can produce them, and polyunsaturated we have to get from our diet, and ask what happens if we repeat this experiment we did with oleate. And here's what happens. Let's take, because I want to do this in the interest of time, I'm going to tell you 2 things. The way free acyl chains get into cells is piggybacking on albumin, and people probably don't know that. Albumin can hold several molar equivalents of fatty acids in its 7 binding pockets. And it's taken up constantly by endocytosis in cells, and then in a pH-dependent fashion, it gives up its cargo back. And so you can introduce a specific lipid by adding fatty acid depleted albumin, and then adding back your favorite fatty acid, which is what's done in these experiments. And what you can see, is that on the right-hand side in complete media, the only way we can grow these fatty acids is if we add them a polyunsaturated fatty acid. It doesn't matter which one we add. The two essential fatty acids that we normally depend on, linoleic acid 18.2 or the other one 18.3, will actually allow the droplets to grow and begin to crowd out the whole cell. So you see on the right-hand side, when we add the LA, linoleic acid, it's the only lipid we can add in complete media that makes lipid droplets in kidney cancer cells grow, and it'll do it even in the serum starved cells. Oleic acid will keep you from depleting the proteins, but it won't actually make them grow. So what makes the size of this thing in serum is actually the ability to have and modify lipids through polyunsaturated fatty acids. Why is that interesting? Because the excitement around the cell death community comes from this recent discovery of ferroptosis, that all cells are susceptible to reactive oxygen species because reactive oxygen species in the presence of oxygen, remember renal cancer is a highly vascular tumor, so there's lots of oxygen normally, despite some regions of hypoxia that you all have reported. Normally, this is a highly vascular tumor. Oxygen will, in the presence of iron as a catalyst, capture single electrons and then do oxidation reactions. And the one that self-perpetuates is a reaction that's outlined, on this slide. I got ahead of myself. Let me just go back and tell you one thing. So the first thing we learned is we did the single ones, is that if we add a monounsaturated fatty acid, and here's the two most common, so this is oleic acid, and this is the 16-1 form, and we can completely rescue cell proliferation, even though the lipid droplets don't really accumulate that much. If we add palmitic acid, the cells still mostly die. So the saturated form of that does not work. It's a monounsaturated fatty acid. So there's something important about that. But if we add the polyunsaturated fatty acid, which makes lipid molecules accumulate, and you get a clear cell-like morphology, then when you release serum, every cell dies. And it doesn't matter if you put it in complete media, they can figure it out and adjust it. So there's something bad about accumulating polyunsaturated fatty acids. That's what we tend to do when we overeat. We tend to have those exogenous fatty acids accumulate in excess of what we normally physiologically do. And so that made us realize this issue of ferroptosis, that polyunsaturated fatty acids have been discovered to be an intermediate between reactive oxygen species and cell death by this process. It's iron catalyzed, where you destabilize membranes by lipid oxidation. And so that's really pro-inflammatory, so it may explain why, even though there aren't many mutations in kidney cancer, it can be inflammatory in a way that is immunogenic and may play out in some of the pathways of obesity that we'll talk about at the very end here. But I want to make one really important point without really going through all the data, because I'm getting conscious that we're a little late on time. This is true of every tumor that we have found from the proximal tubular, okay? So this is not VHL dependent. This is dependent on the phenotype these cells maintain in the NHS. They think they're a proximal tubular cell, and that's sufficient. So KACI1, one of the original ones, doesn't have a VHL mutation, and it still undergoes this same phenomena of oleate-dependent cell proliferation and serum deprivation, and that's what's shown here. And any ferroptotic inhibitor, which is shown for A498-760 and KACI1, will rescue that proliferation independent of exogenous lipids. So they have enough endogenous stores of lipids, or the ability to regenerate lipids, as long as you prevent ferroptotic death. So the reason these cells can't proliferate in the absence of oleate is because they undergo ferroptotic cell death if they're lipid laden and you remove serum, so they can't get an extracellular supply of lipids to build their membranes from. So unlike every other cancer we have studied, renal cell proximal tubular derived tumors cannot engage in sufficient cell autonomous lipid production. They have to live on what you eat for lunch. They build their lipids from what floats by and what's held by albumin. The reason they can't engage in cell mitosis growth is that when they accumulate these lipid droplets, they turn off the major factor that regulates the production of lipids. They turn off SRABP. That's what's shown here. And in a serum-dependent fashion, based on the size of the lipids, we think it's the intercellular lipid concentration, they regulate their cell autonomous ability. So once a cell's full of lipids, it can't reactivate its own endogenous ability to produce lipids. And if you eliminate, though, in this recovery period, the ability to restore lipid biosynthesis by blocking FASN. There are FASN inhibitors that are in the clinic right now for various forms of cancer. These cells cannot recover. They do need some endogenous ability as they recover in this proliferation to go back, like every other cell type, to endogenous synthesis. And if you take out completely endogenous synthesis, because I painted this as black and white, it's not black and white, it's just a suppression of this cell autonomous ability to do proliferation. If you take out completely that ability, they shift by a log their sensitivity to ferroptotic cell death, renal cell carcinomas. So this is just to sort of make a point. All the lipids that are being built in vivo to make a clear cell phenotype actually come from the extracellular environment. So Simon a couple of years ago published a paper that said that these cells can't take up cholesterol. That's true and validated in this experiment. The reason they can't do this on their own is because of the sensitivity to ferroptosis. That's what's shown here in various combinations of giving them back lipids in both cholesterol. Cholesterol by itself plays no role in the ferroptotic process. So if you just look at the line where we've given delipidated serum, which is the 3% LDPS, and no cholesterol. Let me see if I can just, I guess I can't show it. Cholesterol won't rescue. It's only the monounsaturated fatty acids that rescue this. So this is about de novo fatty acid synthesis, but cholesterol makes the cells better under all circumstances. Okay, so is there any data to support what I just said, which is the last thing I'm going to tell you about very briefly? If you look across the entire TCGA and ask which tumors are the least good by gene expression of being able to make their own lipids, it's renal clear cell carcinoma. It's at the bottom. It's statistically lower than all the others. If you actually look, and some of this has been published by Simon, blue is, remember, low expression. The entire cholesterol synthesis pathway is turned off in renal cell carcinoma. So that's already published that. What we added to this is they also have turned off all the de novo fatty acid synthesis pathway as a population, okay? But what they retain are all the genes that the normal proximal tubular turns on during differentiation to scavenge the lipids from urine. So their profile of lipid uptake is all bright for scavenger receptor uptake, which is the way you scavenge it from urine. And the only gene that's down in this panel that I've just shown you is the LDL receptor, which is the normal pattern growth factor turns on, but they don't have receptor tyrosine kinase activation. So they get their lipids, these tumors that you guys work on, from the scavenger receptors. Because there's no urine going by, they're taking it from what the HIF mutations have delivered through VEGF, of the lipids that are leaking out of the bloodstream, and that's why they can, more than any other cell, take up just tons of extracellular lipids from their environment, but they do it through scavenger receptors. SCARB, which is on here, is what Slisha, you may know that there is a megalin complex on the apical side of proximal tubular cells, which is responsible for the major part of scavenging. It brings together this, and this is still maintained on all the renal tumors that we've been able to identify. So that fits this very nice model that based on what you eat, okay, most cancers go to de novo fatty acid synthesis. They make a balanced amount of saturated fatty acid muffin, make really pristine bilayers. But we have these kidney cancers that actually use lipid uptake from our diet. So that's the mixture. So it still has monounsaturated fatty acids and saturated fatty acids, but it's mixed with polyunsaturated fatty acids, and that puts them at risk to death under various perturbations, which we would argue are the things you do therapeutically. And that might explain the lipid paradox. So you can do some experiments in vivo. You can take these tumors that are completely loaded. I've showed you in response to how much serum we can measures how many cholesterol esters in this case, but it's also true of triglycerides. And what you'll see, which is really interesting about the cholesterol esters, and this has already been published, this is just our data set to match this, others have done this, all of the common cholesterol esters, which make up that lipid droplet that makes the clear cell phenotype, are all bound to polyunsaturated fatty acids. And if you deesterify those, they kill cells. They get put back into the pool from which you build lipid bilayers and make you susceptible to ferroptosis. If we take an agent that is just blocks the deesterification, so it's a cholesterol esterase inhibitor, which is just a compound, we can restore in the presence of serum deprivation, the release of these cholesterol esters, and as I think I've got in the next slide, that restores cell death without a ferroptotic inhibitor. So somehow the release of these esters is also perpetuating cell death, either because they're incorporated in the lipid bilayer or just on their own, they're creating oxidant damage around the cell as they're released. We do not know the difference between that. But I have one last piece of data that I want to show you. This just says if we eliminate the cholesterol esterases, the cell becomes incredibly resistant to apoptotic cell death. So if the renal cell carcinoma can't release these cholesterol esters and release the polyunsaturated fatty acids, they're not susceptible to ferroptotic cell death. But this is the data I wanted to show you. This is many of you in this room have contributed to the idea that FASN is a bad prognostic feature. That came out of the genomic gene expression era. And it's a pretty good marker. It's not quite as good as obesity, but if you just take data sets that exist in the public and look at FASN, that's what we've done over again, but we didn't discover this, you all did. FASN is a bad prognosticator. If you can make your own lipids rather than have to take them upside, you have a bad prognosis of kidney cancer. But the reverse is true. If I take that panel of lipids that I told you about, and I do the correlation on survival with the clinical database, you can see it's almost as good as obesity at predicting you're going to have a good outcome. So if kidney cancers are dedicated to only using lipids from exogenous sources, they respond better to your therapies. That's a great predictor as a predictor going. So no I involved here yet, but we need the data sets to be able to do that. And the reason we need those data sets is just this. There's only one data set that combines metabolomics, patient outcome, and gene expression analysis. So we took that data set. It was made by Ari Hakimi and Ed Resnick. And what you can see is if you ask for the ratio, what percentage of the phosphatidylcholine that makes up membranes, this is total cellular membranes, how much of the phosphatidylcholine that makes up lipids is polyunsaturated. It's highly enriched in the tumors that have a lipid scavenger phenotype, and it's very low in the cells that have a FASN or ACA, which is the other essential gene in de novo fatty acid synthesis expression. It's highly statistically significant in this data, even better at predicting the prognosis than any of the other markers that I've told you about, which suggests that this is really about the sensitivity of these cells, because they're lipid scavenger, to this Achilles heel of taking up some things we ate from our diet that actually lead them to be more susceptible to death under stress. And so with that, we need more databases. And we need the power to analyze those databases, and that's why we're going to turn to AI for the rest of the morning as we go forward. Because this is at least an inkling of idea that I wanted to introduce at the start of the community. We've got great strategies that have improved kidney cancer, as we heard from Brian and others going forward, but there's even new ideas that can be put into place as we start to understand these omic databases, and we complement the gene expression, the single cell analyses, with these kind of metabolomic analyses. And so with that, I'm just going to leave you with these conclusions. I'm happy to take any questions, but I realize we're cutting into time to hear about AI, which we're all excited about. So thank you very much for your time.